Let's go to the first one, where we present a genome. So we take genomes, bacterial genomes, mostly annotate them with protein families. So each shape here is a different protein family. And then we construct what is called the phylogenetic profile matrix, which is just a matrix of ones and zeros, which says the domain or this protein is present in the genome, yes or not. Look, for instance, here this square is 2 times, still I have only a 1. So it's only a 1, 0 at this point. And what phylogenetic profiling in the original formulation did is to look to some statistical correlation. So in this table here, one line, like this red one here, is one bacterium, and now a column is one domain. So it's a generalization or a modification of what I had in the multiple sequence alignments. And here you see that these ones, for instance, are they're correlated for 4. It's hard to say what correlation means, but you see some pictures, they co-occur or they are present, absent together, and you can measure if there is some statistically significant correlation or not. And you can say the most correlated are maybe interacting with each other. So what we did, driven by prior experience of this residue scale coevolution, is that we said, okay, now let's formulate this better in some global statistical approach. So let's use the fact that we are statistical physicists in our group and write some model which is a joint probability of one of these red vectors here, which is a 0, 1 vector, over all domains, and write it as a form in a form which allows for the fact that some domains are more frequent than others, and that domains in particular are interacting, they are correlated. And so this is basically a simple Ising model, or here would be a lattice-cast model more specifically, but that's just wording because they are totally equivalent by some simple transformation. So and if you infer this with methods from inverse statistical physics, and there are many of them, and I don't talk at all about technical aspects here, then you can get couplings, so these Jij, and you can construct another network. Maybe better, maybe not. The data we are using is in bacteria, for instance. If we take all bacteria, we had in the data set which we were using 1,041 species and 9,000 domains. And if we match this to a reference of E. coli, so all those which were E. coli is a total 11111 vector, so all those domains which are present in E. coli, we had obviously the same species, but only 2,000 domains. If we look to human, we have less species because in eukaryotes there are less species, but many domains present. So the matrix becomes less favorable from a statistical point of view because we have less samples in higher dimension. Okay, so we do with several methods this inference, and what we find is something like this for the histogram of the couplings, and you see this doesn't look like a Gaussian. So this one is logarithmic here, a Gaussian would be a parabola, but you see that there is some characteristic shape. There's something, actually a huge peak. So just to show you, this one is 10 to the 7, and here we are 10 to the 2. So 5 orders of magnitude only in this central peak, which is very small couplings. So this may be noise, this may be something which is due to phylogenetic relationship between the species, and so something we can't interpret. But we have 2 tails. There's a small tail with negative couplings, and actually if you look into these cases, it's quite funny because you find cases of shared functionality at very low or non-existing homology. So you find cases of known convergent evolution, so where basically evolution has found 2 times a solution for the same function, but you also find cases where actually evolution went so far that now it's characterized into 2 protein families. But there is some common ancestry even further back. Okay, you find some cases, but it's not so much a signal. There's a much bigger signal in this kind of fat tail here, which have positive couplings. So positive couplings, I should have said this also here, negative couplings means if one is present, you should better, the other should better be absent, and vice versa. So, and that's actually the case if you have the same functionality, you just need one of the 2. And if you would have 2, it would be energetically not very favorable for the cell to have them. So there is some reason why, you could expect this. And here you favor co-occurrence, so they either appear together or none of them, obviously in a statistical sense. So you will find cases, but you will find more cases that they co-occur, not occur, co-absence, than one of them being there. So to actually get... I have a problem with... I generally have an ideological problem with flags, and this is really manifesting itself in my incapacity not to step into it. It's okay, thank you. So to get an idea if what we did makes sense, we have compiled a list of positive domain-domain relations, and we have done this in a very large sense. We have done some questions of genomic localization. I told you that domains are building blocks of proteins, so they can be localized inside one protein. They can be localized into proteins which are close by in the genome, in bacteria in particular, and operons. And we have some real interactions which we have compiled from some databases. One is more general protein-protein interaction database. The other is a structural interaction database. And then we have added some metabolic reactions, which is basically if you have in a metabolic pathways two subsequent reactions which are catalyzed by 2 proteins. And then we have compared to our data. So what I show here is the positive predictive value, which is the in between, for instance, the first 10 or the first 100 predictions. So the first, I mean that I start with the biggest one and then I sort by size. So I start with the biggest one, and then now I take the 10 biggest, the 100 biggest. Okay? And I look how many of these are in my database in here. And if you look to correlation measures, you find these curves. If you look to these coupling measures, you find curves which are superior, so which are better because we have more good predictions. A perfect prediction would be this line here, which would have all these 8,000 relations before the non-relations. So we are not perfect, but we get better. And we have very different enrichments. We don't find any signal, any enrichment for the metabolic relations, but we find enrichment both for localization, like the yellow curve here, or interaction, like the blue or the green curve. So we find that there's a mixture of relationships which are real physical interactions, so 2 domains which form a complex, and things which have some more general functional relationship, which we cannot necessarily characterize by protein-protein interaction.
So, Martin, there is no big difference between mean field and pseudo-likelihood methods for Weigt.
Yes.
So it means that the inverse problem you're doing is very simple. It's not a complicated inverse problem.
It depends on what question you ask. In terms of the topology of the predicted interaction network, they are very similar. And that's something which is a common motive to basically all situations we know. But in terms of if you want to have a generative model, which is a question which I'm not addressing at all, mean field is doing a terrible, terrible job. So the mean field couplings are very correlated to the pseudo-likelihood maximization couplings, but they are some factor higher. So it's like going to very low temperature. So it's a disaster for actually taking the model as a statistical model.
It makes no sense to put any regularization to remove small j's, put it to 0. It doesn't help.
This one is not. You have to remove the very big j's, which are due to small sampling. But I can tell you about this. I will never reach the end if I try to go into this detail. Already, like this will be hard. So now, okay, we have considered this table, and I told you that our knowledge is very incomplete about protein-protein interactions. So why to say that all these are wrong? Maybe these are, they have a strong coupling, they have no known interaction, so why not taking them as a prediction? With this kind of data, I'm somehow stuck here. I can look into them and can discuss them and so on, but I can't go on further with the data because somehow I have exhausted my data, which is just a 0-1 presence-absence representation. So what I would like to do is now to complement this by an information coming from this lower scale evolution, which was this co-evolution on the residue scale. So something which is summarized in this picture. So if I have two interacting proteins here shown by these two chains, they have some contact to residues which are one in the first, one in the second protein, and they are in contact. I told you they tend to co-evolve because they have to keep physicochemical properties which are related, and this co-evolution is found in terms of correlated usage of amino acid, like here symbolized by this little totally fake example I have here, where you have R and D together, E and K together, but no RK and no ED. So basically the fraction of sequences using some pair AB is different from the fraction of sequences in the first protein using A and B in the second position. So there is some correlation. So here RD would be 50%, and it's different from 50% times 50%. Okay, and this can be put again into a model which is very similar to what we have seen before. Just now it's a model over the 21 letters which are amino acids or gaps which are in our original sequence alignment. And we have this model which we call direct coupling analysis some years ago, and there is a review, quite complete review, relatively recent, which shows what was done with this model. Basically, these Jij guarantee that this is reproduced. Okay, so we can do this. This method has found quite some applications, and one of the reasons is because the largest couplings now between residues, so not like before between domains, but now between residues, were frequently found to be contacts. This is a picture from results from our first paper on this subject, a picture from a paper by the David Baker's group, and the lines here or here are the predicted contacts, and they are frequently found to be in contact. This in the last years, actually, the field advanced quite a lot into one particular direction, which is due to the fact that these predictions are still quite noisy. So for many proteins, you actually don't find contacts, even if you know they interact.